Okay, let's get started. So, hello and welcome to the third ICMRBS ECR webinar. It's great to see so many of you here, even with the change of time. So we changed the time to try and accommodate speakers from Australia and Europe. And this is something we're quite keen to do, to try and include speakers from all around the world. So on that note, if you'd like to speak, email us. We've got a Twitter, we've got a webpage. If you'd like to join the mailing list, the ICMRBS mailing list, then email Kevin Gardner. Please don't share or record any of the talks today. We're going to upload as many talks as possible to the YouTube channel if we have permission. And please type any questions during the talks into the Q&A session and we'll get to them at the end. So I'm now going to pass you over to Carol, who's going to introduce our first speaker.
Hello, everyone. Today, our first speaker is Lorna White. She has a bachelor and PhD from the University of Sydney. Following her PhD, she spent 80 years as a postdoctoral research with Professor Jack Matos in University of Sydney in the field of protein structural biology. Since 2016, she has helped to set up and manage the fragment-based drug design platform in Sydney University, which has been part of the Sydney Analytical Core Facility since 2018. Lorna's main role is carrying out the fragment-based drug discovery screens for internal research groups, as well as for the wider Sydney research community on a variety of protein targets. These screens are performed using MR and surface plasmon resonance, and she also works with research to characterize protein nucleic acid, and protein small molecule interactions using NMR, SPR, and also ETC, and in silico docking methodology. Additionally, she runs NMR-based metabolomics analysis on a variety of samples type. So welcome, Lorna, and take your time.
Thanks very much, Carol. I'll just do the usual thing and get my screen sharing going. Second. Okay, hopefully everyone can see that okay. Just give me one sec. Okay, so I'll assume you can see it unless I hear otherwise, and I'll assume you can hear me again unless you tell me otherwise. Okay, so thanks, Carol, for the introduction, and thank you to the organisers for letting me come and talk to you about one of my FBDD projects today, which is targeting BRD3 ET. So I'd just like to start with the acknowledgements for this project, just to sort of show that it's a really wide-ranging collaborative effort that I'm going to tell you about today. So it's headed by Professor Joel Mackay at Sydney University at the School of Life and Environmental Sciences, that's SOLS. We've had a few of Joel's grad students work on the project over the years. That includes Dorothy Y, Taylor Sazika, and James Wood-Manzi. All of our NMR infrastructure, or our biomolecular NMR infrastructure, is managed by Dr. Anne Kwan. These days it falls under the same core facility that I work in, which is Sydney Analytical, but in the past it was also managed through the School of Life and Environmental Sciences as well. And in all of those cases, very much ably managed by Anne, and I thank her for her assistance in all things NMR in this project. This is a collaboration with the Monash Fragment Platform at Monash University, headed by Professor Martin Scanlon, and sort of overall my platform works really closely with their platform. We've also had input on the project from Dr. Brad Doak. The medicinal chemistry work you'll see towards the end has been done by Luke Adams, and the SPR that was done at the Monash platform by Menachem Gunsberg. We've had general NMR input from Biswa Mohanty at the platform, and Steve Heddy, who is formerly with the platform, was involved in the early stages of the work. Joel also has collaboration with Professor Ben Capuano from Monash University in the area of medicinal chemistry. And we, of course, thank ARC for support on the project. So, as I said, I'm going to talk to you today about BRD3 ET. BRD3 is a member of the BET bromodomain protein family. There are 4 members, BRD2, 3, 4, and T. They're named for their bromodomains, of which they have 2, shown here. Bromodomains are really well characterized domains. There's a structure of one here on the right in gray, and they're well known to bind to sequences that contain acetylated lysine residues. And again, one is just shown over the top there in yellow. These acetylated lysines occur in things like histones and transcription factors. And so these domains, and therefore these proteins, are key players in transcriptional regulation and control. And what also comes with that is the fact that they're often also involved in cancer pathways. Because of this fact, they're really heavily mined in the drug discovery space. You may well have heard of bromodomains and the various drug discovery efforts people have put into targeting them. There are many molecules in clinical trials. As far as I'm aware, currently none have made it to the clinic, but there are many different molecules through the sort of various phases of clinical trials with the idea of being anti-tumor agents. Now, in the Mackay lab, they are interested in transcriptional regulation and control. But in addition to looking at these bromodomains, which, like I said, are quite well known, they were sort of interested in what's going on with this extra terminal domain here that happens to be at the C-terminal end of these BET family of proteins, which is the 4 I mentioned before. And now what their lab and others have shown over the last few years is that the ET domain in its own right can actually bind to a variety of regulatory proteins. And one of the most famous of those series is CHD4, which is involved or is part of the nucleosome remodeling and histone deacetylation, or NERD complex, which is a key sort of transcriptional regulatory player. Now, these proteins, there's quite a variety of them. They're quite different. However, all of them interact with the ET domain via a short peptide sequence that always contains a KIKL motif. So I'm just showing the structure of one of these here. This is the BRD3 ET domain in surface with the charges highlighted, and shown is the peptide, specifically the KIKL motif. This one just happens to be from CHD4. And it was solved by Dorothy Y in the Mackay lab as part of sort of the characterization work that was published a couple of years ago now. So the question that came from this work was, well, we can see that the ET domain appears to have a role in transcriptional regulation and control. So maybe it's an alternative drug target. As I said, the bromodomains themselves are very heavily mined, but no one had actually really looked at the ET domain. So this is sort of where I came into the work to have a look at things from the drug discovery point of view. And as you may have guessed from where I work, we decided to use a fragment-based drug discovery approach for this. So just as a little bit of background for people who may not be aware of what FBDD is or how we sort of do it, essentially it can work as an alternative to things like you might be more familiar with, such as high throughput screening. So with high throughput screening, you're going to screen more like actually hundreds of thousands of compounds against your target of choice. And you're going to hope to pick up something with a pretty high potency. So we're talking micromolar to nanomolar affinity range. The drawback with this approach, however, comes in terms of chemical space. So these molecules are around 400 or 500 Daltons in size. To cover all the different possible combinations, you'd need to actually screen an almost infinite amount of compounds. So you might come away from a high throughput screen without a hit if you're in the wrong part of chemical space. Also, if the compound is a poor fit, as shown a little schematically here, you're going to need to redesign it anyway in order to get that high affinity, high specificity binder that you're after. So with FBDD, we take these compounds and we break them down into their constituent elements. They're going to be small, so we're talking 100, 150 Daltons in size. And because they're small, they're going to bind weakly, so millimolar to high micromolar affinity. However, screening thousands of fragments, which is pretty doable with today's automation, allows us to cover significant portions of chemical space. So you're much more likely to come away from a fragment screen with a hit or series of hits. However, what you have to remember is that this little fragment is not your final drug, and you're going to have to do some work to actually develop it. So how do you get there? Well, a couple of different ways. If you got particularly lucky, and let's say you got fragments binding in adjacent portions of your binding pocket, you could do some chemistry and link those together. More commonly, though, we have to employ this growing approach. So we'll take our fragment, we'll look at a series of different functional groups, and we might find that this one's good at aiding inhibition. Well, then, as per this example, we might look at targeting the additional portion of the binding pocket, or you may just look at growing your molecule further to sort of aid inhibition. And in the end result, it's going to be the same, which you're hoping to gain a high affinity, high specificity binder. So as I said, this is the general approach we took with screening of BRD3ET. We screened the Monash fragment platform library that we use. As I said, it's been developed, as you probably guessed from its name, it's been developed by the Monash fragment platform. It's a very well curated library. It's designed to maximize chemical space coverage while minimizing not off-target effects, but artifactual effects, things that might come from aggregated or precipitated compounds. It's around 1,180 compounds. We screen the library. We screen using NMR ligand detect, specifically saturation transfer difference NMR. It's a good experiment because it'll pick up the weak interactors that the fragments are going to be. Also, it's a ligand detect experiment. So it doesn't matter if your protein is large or small, because you're not looking at the protein, you're looking at the ligand. With BRD3ET from the screen, we selected 70 hits, and we decided to validate those using 15 and HSQC. So normally with larger proteins, we'll use a ligand detect type of experiment. However, because BRD3ET is quite small, it's around 11 kilodaltons, we could use 15A and HSQC, which just gives us a little bit more information. So we did end up testing all 70 of those hits, but what we picked up on in particular was this fragment here, which appeared to bind pretty well just by even eyeballing the HSQC. So the blue is the protein alone, and the red is plus 1 millimolar fragment. That's around a 10-fold excess. These shifts are really good for fragments. We don't usually see things that are quite this good. So this was pretty exciting. Because the shifts were so good, we could actually even do a titration, which we don't normally do out of a primary screen, but we could. And so we were able to get a KD of around 220 micromolar. The other question, in addition to the fact that it appeared to bind well, was, well, is it binding somewhere on that peptide binding interface? Because ultimately that's what we're trying to target. Just as a really easy and quick first pass, we just compared the HSQC profiles. So on the right here is the profile. Again, blue is protein alone, red is when you add the peptide partner. And just so you don't have to eyeball the entire thing, just to highlight a couple of the residues where it's quite easy to see that in both cases we saw similar shift patterns. And we did some more in-depth analysis of that later on, but just it was nice to see as a first pass that this fragment looked like it was binding somewhere that we were interested in targeting. So as I mentioned, a fragment is not your final solution. We need to go on and start to develop it now. And a big part of that is to start building the structure activity relationship, or SAR. So that just boils down to, how can you grow your fragment? What addition of functional groups work? Which ones don't? Where can you put them? So on and so forth. Just as sort of my visual representation of SAR down the bottom here, these are some of the things that you might actually look at testing, for example. So we did this by what we just call SAR by catalog, which is a fancy way of saying we purchase these fragment analogs. So these, when we start building the fragments, we just call them analogs. And we purchase these rather than synthesizing them. I'll just point out that the Monash Fragment Platform library is sort of designed for this purpose, in the sense that every hit in that library has a series of purchasable analogs associated with it. So it just aids to streamline the whole process. For ET, we were able to purchase and test around 16 analogs that we could get relatively cheaply, and that we could get soluble enough to test in things like an HSQC titration. So as I said, we tested them both by NMR HSQC and also with surface plasma resonance SPR as an orthogonal technique, just to confirm the affinities that we were seeing. So SAR is not about finding something that binds better, it's about looking at the overall trends. So just to show you a couple of those, things that we found were that expansions of this hydroxyl position were well tolerated. We could add quite large additions here and still maintain or get slightly better affinities. The top affinity here, if you're looking at numbers, it comes from the NMR titration, and the bottom one from the SPR titrations where they were done. So yeah, so we could see that adding groups here worked quite nicely. We could also see that small additions off the aromatic ring were also tolerated, be it halogens or methyl groups. However, larger things were not as well tolerated. So perhaps there was some sort of steric hindrance or something going on there. We didn't know, but we knew what not to do. We didn't do a lot with the middle position. However, we did see that it was somewhat tolerated, although it did appear to be quite charge-based. So if you had a sort of a positive charge stuck on the end here, it was fine, and negative charge not so fine. This sort of tracks with what we know about the interaction. If you remember, I said that the peptide binder is through a Ki-KL motif, which is positive. The surface of the protein, if you'd eyeballed it earlier, is quite negative. So it makes sense that something coming in with a negative charge is probably not going to bind particularly well. So that's things on the fragment side. What about on the protein side? Basically, all of the fragments we tested in the analog series all caused pretty much the same chemical shift patterns. So wherever they were binding, they were consistently binding there. We had HSQC data, we have a structure, and we also have the, because of that, we have the assignments. So we were able to map, we calculated the significant shifts, and we were able to map those onto the structure. So here is in gray and cyan is the BRD3ET structure. Purple is the peptide binder, and in orange is the surface where significant shifts occurred. So you can see that it's quite nice that it's mapping to one region of the protein, and secondly, that it's mapping across that peptide interface. So we knew that earlier, but actually mapping it onto the structure was really encouraging. So this is great. This is a lot of good information, but ultimately we like to know at a molecular level how these protein fragment interactions are actually occurring. How is the fragment actually sitting as it contacts the protein? BRD3ET is a great target to work with. We can make lots of it. It's really stable, it's really soluble, but it will not crystallize to save your life. So what we decided to, oh yes, that's the other thing I just want to point out. There are a lot of structures in the PDB for this domain. It's not like there aren't structures, some in complex, some alone, but they're all NMR structures. So I think, again, that sort of tells you something about this domain and how it's going to like work structurally. So we did decide to use an NMR approach with this domain while trying crystallization sort of in the background. We've had a few grad students try it. We have not had any success, but we've at least been able to use NMR to gain more information as we sort of went along. So we decided to start out with looking at a structure or gaining some structural information with one of our better binding analogs. So what I did was use NMR to derive a series of NRE restraints for the BRD3ET fragment complex. And then rather than doing the full structure determination route, which can be pretty sort of laborious and time consuming, especially when you're talking about a complex, I just use Haddock to obtain a molecular model. So Haddock is a really good docking program because it allows you to input a whole lot of different restraints based on whatever you've calculated. What I ended up using was soft restraints, which were basically the residues that were causing significant shifts in the HSQC, as well as a set of hard restraints, which were the NOE distances that I'd calculated earlier. So that just allowed me to sort of position the molecule a little bit better than if we just said, well, here are the residues that shift and here's the molecule. So Haddock will give you an ensemble of structures. And as I said, it's a molecular model, not an actual structure. When we looked at the 20 lowest energy structures, they all had the same pose of the molecule. And just for sort of ease today, I'll just show you the lowest energy structure. Just a quick little video. There we go. So what you can see is that the analog or the fragment is sitting quite nicely into this binding pocket here. Fragments really do like pockets, and it's positioning itself this way with the sort of the smaller expansion outwards and the larger expansion inwards here. Just to zoom in to show you what sort of the contacts are. As I said, it parks itself this way into the protein, and it makes hydrogen bond contacts to the nearby isoleucine 616 and 599, and also to the valine 596 here on the adjacent helix. As I said before, expansions off of that original hydroxyl here were well tolerated, probably because they are able to sit into the pocket and make additional hydrogen bonding contacts. So in this case, this guy was making, again, contacts to 616 and 614. And I guess the other thing we looked at was to do with the peptide as well. So again, probably from what you saw earlier, you guessed that it is sitting somewhere across the peptide interface. And again, that was the case. The KIK motif sits across here. So we sort of had our fragment exactly where we wanted it. So that was all really encouraging to look at. And again, a little bit quicker than going through the full structure determination process, but still giving us good information as we move forward. Just change my slide. There we go. Okay, so we had a lot of SAR information and we gained it relatively quickly because we'd purchased things rather than having to go through the synthesis process. So just sort of this here is just a quick summary of the SAR that we gained overall. However, despite all of that information, which is really valuable, we were still in the hundreds of micromolar range as far as our KDs went. So we still don't have a drug. We're still in the early stages. So what we needed to really do now was to start to use medicinal chemistry to actually improve our molecules into a more drug-like space. So this part in particular has been a collaboration and mostly the work from the Monash fragment platform, as I said, headed by Martin Scanlon. And a lot of this work has come from Brad Doak. And the workflow that I'm going to talk about is a lot of the things that he's developed. So if you have questions, I'll do my best to answer them, but please know that this is really his baby, not mine. And the medicinal chemistry I'll show you has all been done by Luke Adams. So the idea behind this is basically that you'll use your SAR to prioritize vectors of interest. And for us, that was initially, was looking at the hydroxyl here. Just going to go back a bit. There we go. And the idea is that you will be able to utilize a variety of different reaction chemistries along that vector of interest in a 96-well plate format. So just as an example, you can change your starting molecule slightly to get the right handle here. Then you'll perform an alkylation reaction in each well of the 96-well plate. And so what you'll get in each well is 96 different functional groups at that site of attachment. So just as an example, I didn't put 96 on the slide, but these are a couple of things that you could have here coming off your R group there. So in the plate, the reaction is confirmed using mass spec to make sure that you've got some product present. And then you basically take that plate and you screen it straight away using SPR and specifically utilizing off-rate screening. So the idea is to make this workflow as efficient as possible and to limit things like purification of compounds and things like that. So the idea behind using SPR off-rate screening is that when you're screening your fragment hits, which are pretty weak binders, you're going to see traces that look like this, with a very fast on and a very fast off rate. So similarly, in your plate, the starting materials are going to be much the same, and they're going to have quite a fast off rate. However, the products you've generated, if they do actually bind stronger, which is what you're after, you would hope that they would have a much slower off rate as shown here. So this is when you're screening, let's say, a plate of compounds. Most of them have quite fast off rates, but you can pick up quite nicely here one that has a much slower off rate. So this is obviously looked at in a little bit more detail. This is just to give you the most sort of obvious example. Basically, then you take anything that looks interesting, you resynthesize it, repurify it, and then the binding is again assessed by SPR. And so hopefully what you'll end up with is a trace that looks like this with the nice slow off rate that you're expecting from a better binding molecule. So just to show you from one of our reactions, Luke did try a whole lot of different chemistries over the last sort of year or so. Alkylation, again, the one I used as an example, worked quite nicely for us. So from a plate of 92 compounds where he was able to show the presence of product, he was able to gain to see 3 molecules, they look pretty similar, that had KDs in the around 8 to 10 sort of micromolar range. And those that come from the repurified compounds, not from the crude mixtures. Just as something to sort of to finish off with that was quite neat, he was able to sort of do some chemistry on the chemistry, if you like, basically doing some chemistry off of our expansions, and was able to see sort of some nice, let's say, what about roughly sort of 4 to 5-fold improvements again, just by sort of doing some additional work here to sort of grow the molecule a little bit more. We were hoping to get under sort of micromolar, but we're pretty close. And sort of these improvements for us are still really great from where we started off from. So just to sort of summarize all of what is a lot of work in one very small sentence, the use of this parallel synthesis medchem has basically allowed us to get a more than 150-fold improvement from where we were with our purchased analogs, and significantly faster too than if you had to go through and synthesize each of those compounds individually and test them, one by one. So for us going forward, basically our next question is, we have some pretty nicely binding compounds now in an in vitro sense, but now can we start to put them into in vivo assays? So can we get them to actually get into cells? And if we can we start to sort of probe how this ET domain is actually working in cells, and are these compounds affecting it? And all those sort of questions. So that's really our next step. So I will stop there, and I'm happy to take any questions. Thank you.
Thank you, Lorna. We have time for 1 or 2 questions.
Perhaps I'll just start while people are thinking. It was cool, Lorna. It's nice to see how a tiny little fragment causing all these a pretty extensive chemical shift perturbation on the protein.
Yeah.
Well, it looks like that it has got, if not more, perturbation than the peptide itself. Do you expect
what's up with that? Yeah.
Do you expect a lot of conformational changes upon binding? And our second part of the question is the fact that you expect that the 2 molecules to be binding at the same site. Have you done competition sort of assay to confirm that?
Yeah, the first part of the question, yeah, we don't normally see fragments doing quite what they do in that particular HSQC. We're used to seeing some small shifts, not these quite large things. We suspect it is structural rearrangement. The loop, so you've got the peptide and you've sort of got a loop where they're interacting. That's a bit flexible. So we suspect that when the peptide binds, similarly when the fragment binds, everything moves around a little bit, and that's sort of what you're seeing a little bit, why one small fragment causes such a big shift. So I think there's an element of structural rearrangement going on there certainly. We never did the competition experiments. I was always keen to, and we never quite got there as far as the competition experiments go. It is something we do routinely, and I've done on other projects, but we never did actually get there with this one. But we certainly have, we have things that bind quite strongly, and going forward, it wouldn't be a bad idea to try that with some of our quite strong binding things. Yeah, the peptide binding is around micromolar, so for some of the better looking things, it would be interesting to try. Yeah.
Okay.
I think there's a question in the chat, Carol.
let me click it. Here we go. How long is it stable during an NMR analysis? It's very stable. So both the NMR structure I showed you before, which is the BRD3-ET with the CHD4 peptide, and we did another structure as well in the lab. Obviously, NMR structural data is collected over a period of sort of, days to weeks. Usually only 1 or 2 samples were needed to collect all of that data. So it was really stable over quite a long period of time. The buffer is, now I'm going to have to remember, it's 20 millimolar MES, 150 millimolar salt with some TCEP, basically, about pH 6.5. So nothing particularly special, but it just seems to be, it's just a really small, soluble, stable domain, which is great for everything except crystallography, basically.
One question about if you have information about the flexibility of the protein in the binding pocket, and would that facilitate the development?
We haven't ever looked, not to my knowledge, we haven't done a lot looking at the dynamics. So we've focused on more just growing the molecule and seeing if we can improve affinity. We haven't had an in-depth look at any of the dynamics around that loop or anything like that. I think we sort of more focused on, well, if we can grow the molecule and we can see improvements in affinity, that's sort of where we're going to take our focus. For the moment, we didn't have a lot into the dynamics of that loop.
Okay, thanks, Lorna. Really nice talk. Thank you.
Just stop my screen sharing.
We can go for the next speaker.
There we go.
Yeah, cool. Thank you, Lorna. Coming up next, we have Stefan Nebl from Monash. So Stefan has completed, he's completing, he's a PhD student, completing his, he completed his undergraduate and honors in pharmaceutical science at the Monash Institute for Pharmaceuticals, Monash Institute of Pharmaceutical Science, MIPS. So he's now in his 3rd year PhD in Professor Martin Scanlon's lab. And his PhD project focuses on using solution NMR for structural and dynamic characterization of a bacterial enzyme. So it's all yours.
Thank you. Thanks for the introduction, and a big thank you to the organizing committee for letting me speak. I was very much looking forward to it. So my name's Stefan Nebl from MIPS in Melbourne, Australia. And so my PhD project is generally utilizing NMR in order to derive or give us information that we could potentially use in small molecule drug development. And specifically, I'm interested, as Yanni said, in characterizing both protein structure and dynamics, and trying to work out how we can use that information in tandem in small molecule drug development. So quite luckily, Lorna has given a lovely introduction into fragment-based drug design. So I'll just touch on this stuff briefly. So as Lorna mentioned, we have a fragment library of around 1,000 to 5,000 fragments. They're generally very small and simple molecules, around 100 to 200 Dalton. And the libraries are designed specifically to offer efficient coverage of chemical space. And so we use a range of biophysical techniques to screen our fragment library against any target that we're interested in my case, bacterial proteins, in order to identify hits. And as Lorna mentioned, and as you've seen, typically due to their small and simple nature, the initial hits are very weak, usually in the micromolar to millimolar range. So then when possible, we would like to derive some structural information about the complex of our fragments with our targets. And we generally do this by X-ray crystallography. This structural information gives us ideas on how we can either link fragments or grow fragments or generate SAR by analog in order to come up with a much more drug-like compound. And this structural information is really fantastic, but it's still an incomplete picture. And this is obviously because all molecules have undergo dynamic conformational changes, and especially biomolecules undergo conformational changes at a range of different timescales, reflecting the size of their motion. And so a lot of these dynamic conformational changes are responsible for a number of biomolecular functions like protein folding and protein catalysis, and of particular interest to me, modulating ligand binding. And it's a little less obvious how information about protein or biomolecule dynamics can be utilized in small molecule screening, but as I hope you'll see, it can provide crucial information in developing small molecules. And obviously, NMR is the prime technique that we choose to use. There are a range of well-established NMR techniques that give us access to a broad range of dynamic timescales and give us information at atomic resolution and near physiological condition. So I'll just introduce the proteins that I've been working on, and they are DSB, or disulfide bond oxidoreductase enzymes. And they're a family of enzymes found in gram-negative bacteria that essentially introduce disulfide bonds into secreted proteins. So here's just a simplified model. When a polypeptide chain is synthesized, it's secreted into the periplasm, which is an oxidizing space, and the cysteines exist as free thiols. The first two proteins, which form the oxidation pathway, DSBA and DSBB, are responsible for introducing disulfide bonds between the free thiols to produce a folded protein product. Because though DSBA works in a sort of unbiased manner, it can introduce incorrect disulfide bonds. And this is where the proteins of the isomerization pathway come in, DSBC and DSBD. And they just correct incorrectly formed disulfide bonds to produce this final correctly folded protein. In a lot of cases, this final protein is a bacterial toxin or some bacterial adhesin or something like that. So it's desirable to generate inhibitors of any of these DSB enzymes to prevent the production of these. And any inhibitors of the DSB enzymes have the potential to be developed into antibacterial or antivirulence agents. And so I'll be talking about two examples that I've been working on, DSBA from Burkholderia pseudomallei, BPSDSBA it's called, and DSBD from N. meningitidis, or NMDSBD. So I'll start with BPSDSBA. So prior to starting this project, there were no known structures of anything bound to BPSDSBA, no known small molecule inhibitors. And the work that we've done to try and address that was published in JBNMR earlier this year. So Burkholderia pseudomallei is a gram-negative human pathogen, and notably strains of Burkholderia pseudomallei where the DSBA gene was deleted have been shown to be attenuated in mouse infection models. As a result, this is an attractive drug target in the development of antibacterial drugs. So as you can see, BPSDSBA is a fairly small 22 kilodalton protein with two domains, a thyrodoxin domain shown in blue and an alpha helical domain shown in pink. And within the thyrodoxin domain, there is a pair of catalytic cysteines shown here that are responsible for the redox activity of this protein. Now, if we zoom out and compare the surface to DSBA from E. coli, shown on the right, you can see their active site colored in yellow. And in red here in E. coli is this extensive hydrophobic groove, this pocket where the partner protein DSBB binds. And in the case of E. coli DSBA, a number of small molecules have been observed binding to this region. However, when talking about BPSDSBA, it lacks any well-formed hydrophobic groove in this region. So it was kind of unknown where the partner protein bound, and there were no structures of any small molecules binding to this. Unfortunately, BPSDSBA is a fairly featureless protein. It's sort of like a blob. There's not really any pockets that anything can obviously bind into, which makes it very challenging when dealing with fragments. And so we tried to identify anything that would bind to this by screening our internal fragment library of around 1,000 fragments and came up with 110 validated hits of them. This was one of the top. Through generating some analogs, we came up with this molecule here, I'll call phenylthiazole, which showed some improved affinity over the initial hit and some improved solubility behavior. So here I've just plotted onto the structure the chemical shift perturbations from the HSQC. And as you can see, they seem to cluster in this sort of surface region up here. Again, no obvious pocket, but it is in direct proximity of the active site cysteines. So this was quite promising. When we measured the affinity by measuring the chemical shift perturbations, we came up with a KD around 500 micromolar, And yeah, that's quite weak, but unfortunately not surprising when dealing with such a featureless protein and such so early on in the fragment process. Quite promising though is that our compound showed some concentration dependent inhibition in a peptide oxidation assay. So this being the only real route forwards, we decided to proceed. So unfortunately, given its weak binding, we were unable to generate a crystal structure of this complex. So we opted to generate an NOE-based NMR model using haddock as Lorna kindly described. So here again, I've shown just the lowest energy conformer. As Lorna said, haddock generates an ensemble of structures. This is just the lowest energy conformer. And as you can see, it seems to be interacting in this previously unidentified sort of surface area here in direct proximity of the active site. So when we take a look at a crystal structure of a peptide derived from the partner protein DSBD that was released in 2018, we can see that the partner protein seems to interact in this region here. And this site from other DSBA homologs, we have evidence that this may be the substrate binding site of DSBA. And what's quite promising to us is that our compound phenylthiazole here seems to be overlapping in its binding site with this putative substrate binding site, suggesting that our compound may be able to competitively block substrate binding. So up until then, we had only really characterized the interaction between phenylthiazole and oxidized BPS-DSBA, but we also wanted to know whether it bound to the reduced form of DSBA. So we re-recorded the HSQC and we found that yes, it does interact in generally the same site. We see the same chemical shift perturbation pattern, but to a much, much weaker extent than when it's oxidized. And this was quite puzzling to us. We didn't know why there's this difference in binding affinity if the structures are very, very similar, almost identical. And so in order to understand that a bit better, we actually measured the backbone amide proton CPMG relaxation dispersion. And what we found was a number of residues, 36 residues showing dispersion in the oxidized state. So as you can see here, I've plotted onto the structure the REX as a gradient to red. And they seem to cluster in this sort of region here where near the active site, near the binding site of our compound. And so this seems to suggest to us that there is some redox state dependent conformational exchange that exists in the oxidized state of the protein, but not in the reduced. I should repeat that when we repeated the CPMG dispersion experiments on the reduced protein, we saw no dispersion whatsoever. So this is sort of where the project stands now. We were able to derive the first model of something bound to BPS-DSBA and characterize some of the dynamics in proximity of this binding site. Again, I've had to fly through this, but if you'd like any more information, please have a read of the paper. But yeah, hopefully, well, this does offer us a path forward for this project at least. So now I'll pivot and talk about the next example, DSBD. And in contrast to DSBA, in this case, we had some evidence of some pre-existing dynamics within this protein that we've tried to utilize in our small molecule drug design. So DSBD, as I mentioned before, is involved in actually correcting non-native disulfide bonds that are formed by DSBA. Key to us is that activity of this protein, DSBD, has been shown to be essential for the activity of nicerial species. Thus, generating an inhibitor of DSBD could lead to the development of new anti-nicerial drugs. So as you can see, DSBD itself is a membrane-bound peroplasmic protein with 3 distinct domains: this transmembrane domain, a C-terminal domain, and an N-terminal domain. And each of these domains contains a pair of catalytic cysteines that are responsible for their function. So the protein functions through a sequential cascade of thiol disulfide exchange reactions, starting with reduced cytoplasmic thioredoxin. This reduces the transmembrane domain, which in turn reduces the C-terminal domain, which in turn reduces the N-terminal domain, and that generates a reducing potential that leads to the isomerization in downstream substrates. So in order to understand the function of DSBD a little clearer, some of our collaborators, namely Begonia Heras and her group at La Trobe, did some work on this. And one of her PhD students, Roxanne Smith, released a paper in 2018 where they characterized the interaction between these two domains. And this got made the cover of JBC. And so in their work, they were able to separately express both the C and the N-terminal domains in order to characterize their interaction by NMR and by crystallography. And here I've just shown on their structure in red the site of their protein-protein interaction. And so the key finding from their work was that the interaction between these two domains, the reaction between these two domains, is completely redox state dependent. So only when the C-terminal domain is reduced and the N-terminal domain is oxidized is there any reaction, a fast forward reaction, transferring electrons to the oxidized protein. However, when the redox states are reversed, there's no backward reaction whatsoever. They also reported that both of these domains have an almost identical redox potential, which raises the question as to how this reaction is unidirectional. And most importantly to us, is this something that we can leverage in our design of inhibitors of this interaction? And so taking a closer look at the crystal structure of the oxidized N domain, they found that there were multiple molecules in the asymmetric unit. In 5 out of 6 of these molecules, this phenyl cap, so-called phenyl cap here, was in this closed conformation. But in 1 out of 5 of the molecules, this phenyl cap was completely open, exposing these catalytic cysteines. In contrast, in the reduced protein, this phenyl cap was observed only in a closed conformation. So this led them to develop a model, a hypothesis that when the protein is oxidized, this phenyl cap region is able to open and close, allowing for interaction with the C-terminal domain. But when the protein's reduced, this phenyl cap is shut closed, preventing the backward reaction, and can only open via an induced fit mechanism with some downstream binding partner. So when we took a look at the HSQCs of this protein in the oxidized state, we noticed that there were 2 residues, namely alanine 99 and tyrosine 105, which showed significant line broadening. And notably, both of these residues directly flank the active site catalytic cysteines. So this was evidence to us that there may be some microsecond, millisecond exchange going on. Notably though, no residues within the phenyl cap showed any line broadening, which indicated to us that if there is dynamics in this region, it must be happening at a range of different timescales, and we would need to characterize it as such. So in order to probe dynamics on the fast picosecond nanosecond timescale, we recorded a backbone amide heteronuclear NOE, and this is the data for the oxidized protein. And what you see here in the phenyl cap loop residues, there is a dip in the heteronuclear NOE, and the same in the active site, this dip here. I've just plotted them onto the structure here. This indicates to us that, yes, this region is indeed flexible on the picosecond nanosecond timescale. Most notably, when we repeated this experiment for the reduced protein, the residues within the phenyl cap and the active site both returned to their average, indicating that this region is less flexible on this timescale when the protein is reduced. So this, again, supports the hypothesis that when oxidized, this cap is able to open and close, modulating the interaction with the C-terminal domain. So to characterize dynamics on the microsecond to millisecond timescale range, we measured both backbone amide nitrogen and proton CPMG relaxation dispersion. And what we found was in the nitrogen dispersion, we found 6 residues, 3 in the phenyl cap and 3 in the active site, showing weak dispersion profiles. This was only present in the oxidized protein state. When the protein was reduced, the dispersion profiles were flat, indicating that this dynamics was not present in the reduced state. So we then measured the backbone proton CPMG dispersion and found 5 residues, this time one of the key phenylalanine residues showing dispersion, and 4 within the active site showing dispersion. And again, these 2 residues here, alanine 99 and tyrosine 105, showing strong dispersion, explaining why we see line broadening in the HSQC. Again, when we repeated this for the reduced state, there was no CPMG relaxation dispersion. Again, supporting the hypothesis that there is only dynamics in the oxidized state. We are going to record the CPMG data at a range of temperatures and a range of magnetic field strengths in order to properly quantify the exchange parameters. But the data that we do have at the moment all fit to a single 2-state global model, indicating that the dynamics that we see in the phenyl cap and the active site is concerted. So, as I mentioned, there is no evidence of dynamics on this timescale in the reduced protein state, suggesting that this dynamics is modulating the domain interaction when the protein is oxidized. So I have to briefly mention this paper, which came out just recently in eLife by Stelzel et al., where they characterized the dynamics of DSBD in E. coli this time. And what they see is largely similar to what we see. In the heteronuclear NOE, they see a dip in when the protein is oxidized, but not when reduced. And they also observe nitrogen dispersion for 8 residues within the phenyl cap. And basically their data is largely consistent with what we see for NMDSBD, suggesting that these dynamics are a common feature in DSBD enzymes and thus important for their function. And yeah, again, they observed no dispersion when the protein was reduced. So we wanted to try and leverage this information in our fragment screening to develop a small molecule that could potentially bind into this dynamically forming pocket. So we screened our internal fragment library, over 1,000 fragments against both oxidized and reduced NMDSBD. We even ordered analogs based on the top hits, but really we weren't quite as lucky. This protein, the dynamically forming pocket is just either not populated enough on that we can access it with our fragments, or our fragments just aren't the right chemical structures to get in there. And what we see is just very, very weak interactions, very small chemical shift perturbations, and that all of these seem to cluster at the complete opposite end of the protein in a region where we have no evidence that there's any functional relevance. So a bit disheartened, but we pivoted to actually screen a different library that we have of covalent warheads. And this is a library of 35 highly reactive chemical warheads that were designed specifically to label nucleophilic side chains in proteins, including cysteine. And so what we found was quite luckily that one of these covalent warheads formed a highly stable complex with reduced NMDSBD that stayed stable over a number of weeks. And so we were able to assign the backbone of this protein, this complex, and find that it seems to bind specifically to the active site catalytic cysteines. And so some preliminary analysis of the dynamics of the complex that we've got suggests that the complex reflects the dynamics of the oxidized wild type protein. And we're at the moment attempting to generate a crystal structure of this complex. So while this isn't exactly what we expected, it still offers a very vital starting point in the development of a small molecule inhibitor of this protein. So hopefully what I've been able to convince you is that we can utilize NMR to provide us with a holistic understanding of both protein structure and dynamics, and that holistic understanding can be useful in small molecule design. So I'd just like to say a massive thank you to my supervisors, Martin Scanlon and Bishwa Mohanty, for their constant support and letting me do this amazing project. My panel members, Ray Norton and Paul Gooley from the University of Melbourne, for their support and good advice. Members of the Scanlon Lab for their help with protein production and teaching me everything I know. And our collaborators at La Trobe University for their help on DSBD, and Griffith University for their help on BPSDSBA. And thank you very much for listening. I'd love to take questions.
Cool, thank you, Stefan. So I can see that there are a couple of questions on the Q&A, so I'm gonna read it out too. So the first question is by Fabio Almeida. I suppose this question is related to the HEDAC model that both you and Lorna use. So how reliable are HEDAC models to design new lead compounds?
That's a very good question. If you are unable to generate any other structural model, crystallize the protein or whatever, if this is all you have to go off, it's better than just a computational docked model, more reliable because it's data driven, right? We feed it both NOE constraints and chemical shift constraints, but really the accuracy of that model is dependent on the data that you feed it. If you have 100 NOE constraints, it's going to be highly reliable. But in our case with fragments, generally we only have a few unambiguous NOE restraints. So you can't rely on the pose that you get, but as a general indicator of binding location and binding pose, it's good enough to go on. It's better than nothing.
Cool. The second question is, given that the BPS-DSBA small molecule binder only partly overlaps with the bound peptide, is there a chance that you could extend the small molecule binder along the peptide path to increase the binding affinity? And are you exploring this possibility?
Yeah, that's a fantastic question, and that's an obvious vector that we would like to expand upon. I won't be involved in that, unfortunately. I'm not a synthetic chemist, and my PhD is coming to an end soon. But that's definitely an option that we are thinking of exploring. And additionally, we're looking at possibly screening our covalent fragment, a covalent warhead library, to see if we could find anything that links to the cysteines and then link that to the fragment that we already have. That's another option.
And the next question is it possible to do SPR with your covalent warhead fragment to confirm that the binding is covalent or and irreversible?
That's a really good question. My knowledge on SPR is quite limited. Yeah, I'd have to ask someone else in our lab about that. But yes, I'm sure that's something that we could do.
Could be on, but it wouldn't come off, I suppose. Maybe it's on an SPR.
Yeah, I'm not entirely sure, sorry.
Actually, in the interest of time, I think we should move on, but then we still have quite a few questions on the Q&A. Can I encourage Stefan to answer each of your questions in the chat, and we'll move on to the next speaker, if that's okay.
Sounds good. Thank you very much.
Great. So our next speaker will be Marta Carneiro. Marta received her BSc degree in molecular and cellular biology at the Universidade Nova de Lisboa, Portugal. In 2010, she joined the MSc PhD molecular biology program at the International Max Planck Research School in Göttingen, Germany. Oh, I've lost the bio. We're back. She conducted her doctoral research in Christian Griesinger's group, focusing on studying protein dynamics by NMR. In 2016, Marta joined ZooBio, where she's been applying NMR-based structural biology to support fragment-based drug discovery programs. Since 2019, Marta has headed the structural biology group at ZooBio, which combines NMR spectroscopy with X-ray crystallography to support the evolution of fragments into leads. So thank you very much, Marta. The floor is yours.
Great. Thanks, Nick, for a kind introduction. And thank you all the organizers for the opportunity to present here today, and really for the great selection of talks and speakers, not only of this session, for which I might be biased, but of all the previous sessions and future, I'm sure. Let me start with a couple of words about ZooBio. As I can imagine, not all of you will know us. ZooBio is a service provider in the area of fragment-based drug discovery. And NMR is actually one of our core technologies. The company have been founded based on NMR screening technology. I will not talk about NMR screening today, but I will actually focus on, let me see, there we go. I would like to focus a bit on the process that actually both Lorna and Stefan already very nicely elucidated with really interesting examples that happens after you screen. That is, you start with your screen hits, and here in principle it can be of any modality, but we also work in fragments. So these are pre-weak fragment hits, and essentially these molecules enter a cycle in which we test them in a variety of assays, and here emphasis on variety. All the assays have advantages and disadvantages, and really the best way of keeping you from going down some rabbit hole is to combine different assays in such a way that each assay covers the blind spots of the other assays you are using. Once you test your compounds, you get some data out of your assays, and you start analyzing them. And this is what both Lorna and Stefan also mentioned, this extracting these structural activity relationships. That is, you try to relate some functional readout that you get out of your assays to chemical features in your molecules. Once you extract some relationships out of this assay data, hopefully you'll come by some nice structural information, and you start designing and making or buying essentially your next round of compounds, your analogs, which will be some variants of your original hit. Once you have your next set of analogs, you essentially enter the cycle again. And the goal or the expectation is that in each round of this cycle, you'll increase the potency of your compounds, you'll improve your compounds until eventually you reach a lead-like compound or a compound series. Among many other properties that you will want for these compounds, one key property is that it should elicit some sort of relevant effect at concentrations at sub-micromolar concentrations. And also very important in drug discovery, you do want to get from these high micromolar to low millimolar compounds to a sub-micromolar compound as soon as possible. Now, what I want to focus in this talk is actually a couple of questions that are often taken for granted within this process. But it's not always the case. And it is good to be aware of them. And even better, it is good to test them. So starting really at the point where you test your compounds, we all assume that the compound we are putting in our assay has the structure that we expect, the structure that we picked and designed carefully. And it's often assumed that the compound is soluble in the assay buffer. But is it really always? And once you start testing your compounds and you'll start deriving all sorts of conclusions and relationships between a certain biochemical or biophysical effect and chemical structures, it's worth considering, is the effect really coming from the compounds? And is the effect I'm measuring a productive effect? An easy example here is a compound that precipitates your protein will certainly be a very effective inhibitor, but it's not exactly the type of inhibition that you can progress into a drug-like molecule. And finally, I already alluded to this in the previous slide. Once you start designing your compound, knowledge of where and how the compound is binding can really make this process much faster and efficient. So what I wanted to show you today is a couple of examples scattered through different projects in which we used NMR, and I really hope to show you how useful NMR is to answer these different questions in a very timely fashion that enables fast progress of the projects. So let's start with solubility and integrity of compounds. Both Lorna and Stefan already mentioned surface plasmon resonance, SPR. So it's also nice to see in this collection of 3 talks that you really see that fragment-based drug discovery people always come around to the same techniques, which speaks highly of the techniques. So SPR is really our workhorse once we have to determine the affinity of several compounds in multiple cycles. And this is an example of binding isotherms for 2 compounds from the same chemical series. Their affinities were determined by SPR. Compound 1 has a KD of roughly 200 micromolar. Compound 2 has a very exciting 5 micromolar KD. Now, you'll be suspicious if you know 1 or 2 things about SPR, about the low relative response. But if you are working on a project and you see this KD, you are not going to discard this compound based on the response alone. You'll certainly be wanting to get a couple of analogs and test these compounds and see if the effect is consistent with a couple of analogs. But now, before you spend a couple of hours or even a couple of days buying more compounds and testing them, it's very worth to do a very quick 1D NMR experiment on these compounds. So for compound 1, we did so, and we see the compound signals that we expect. We know the structure of the compound, so we can assign these peaks and assure ourselves that the compound is intact. And this solution is done in an aqueous buffer, so essentially in the assay buffer, in the presence of a control or a reference compound. We often use TSP. So essentially what we do as a next step is we just integrate our compound signals, and we compare the integrals to the integrals of the known concentration of TSP. And we have a quick assessment of what is the concentration of this compound in the aqueous solution. When we did this for compound 2, well, it turns out that the compound is essentially brick dust. It's absolutely insoluble. We cannot see the signals of the compound, which means that essentially this KD is absolutely meaningless. And I did mention already that these two compounds were from the same chemical class, and I really just want to impress upon this topic. So for this particular chemical class, we had a little over 200 analogs, and indeed more than half were well soluble above 400 micromolar, which is a concentration range we're using in this experiment. But nearly 1/4 of these compounds were not soluble even at 100 micromolar, meaning that any assay that you use concentrations above 100 micromolar for these compounds, you should be extremely suspicious of the results. Now, in keeping with the topic of controls, so what we're talking about here in all these experiments is we're characterizing interactions between protein and small molecules, fragments in this case. These are often very weak interactions in the micromolar to millimolar range. And you've seen in the previous topics, in the previous talks, using something like 1 millimolar of a compound is really not an odd concentration. So you add your compound to your sample at 1 millimolar. It's worth thinking, what else are we adding? Do we have metal ions, salts? Is the compound affecting the pH of my sample? Certainly some of these parameters are very easy to, sorry, I'll just add the laser pointer. Very easy to test, right? You can just measure the pH of your sample with and without compounds. Assessing whether there are metal ions or salt contaminations can be a bit trickier, but just having this idea in mind, being aware that this could be a situation can already help you along the way. Now, what we are certainly heading when we add 1 millimolar compound is hundreds of millimolar of DMSO. DMSO is essentially the ubiquitous solvent for small molecule work. It's often considered to be inert, but it's often also not the case. This is an example from a different project. We're doing quite some NMR work on this project. It was a very NMR-friendly protein. And what we realized very early on the project was that this protein also really liked to bind DMSO. You see here is essentially a DMSO titration starting from apoprotein to increasing only DMSO amounts from 2% to 6%, which are also not odd values to use in such a campaign. More importantly, and I don't show you here, we did have the assignments for this protein. So it also became apparent that these shifts we were observing deriving from the MSO actually mapped to the site that we're aiming to target. Now, this is obviously not what we would wish for a project, but this is also not a showstopper, especially if you realize this very early on. You will know that you are essentially running your experiments in a competition mode, because now you have the MSO, which is definitely a very weak binder. We can also guesstimate KD from this titration, but that is present at hundreds of millimolar concentrations against your also very weak fragments. So what you can do with this information is you can consider very early in the project if you need to use alternative co-solvents, for example, make your compound stocks in methanol instead of DMSO. If you need to use higher stock concentrations, therefore minimizing the amount of DMSO you add to your samples. And you know absolutely that you need to keep the amount of DMSO constant in your titrations and throughout your all your experiments, really. This, I mean, we're all NMR spectroscopists here, or at least the vast majority. We know that it's relatively fast and easy to measure a couple of HSQCs of a protein if the protein is NMR friendly. But this is the kind of effect that you would not easily pick up if you're running your project only on SPR, for example. So it's not necessarily that you have to run all your project on NMR, but it's a very good technique to pick a couple of these quality control aspects. Now to move to a bit of a higher resolution or a more assay-centered view, we move to structural biology. I think both Lorna and Stefan really highlighted the need for structural information if you want to evolve your fragments. And I just want to impress that X-ray crystallography is the method of choice for structural biology, also for fragments. But even there's a little help that NMR can do, even for proteins that crystallize very well. So in this project, we had a 85 kilodalton protein. We had pretty high resolution maps available. But the challenge at this stage of the project was to confidently assign the electron density to small fragments, max 300 daltons, some even smaller. These fragments are also weak, so you might expect only partial or incomplete occupancies. And you want to do this for multiple fragments and as fast as possible. This picture actually comes from that project, and essentially all the red arrows highlight electron density blobs where our fragment of interest could potentially be assigned to. Now, of course, you can do this iteratively and just try all the electron density blob options. But this just becomes time consuming, especially if you have several dozens of compounds that you want to process or structures that you want to process in a timely fashion. So what we did with this project was actually we combined a ligand observed NMR experiment upstream of our X-ray trials to focus which compounds we want to put into our X-ray trials. So in this experiment was actually a T2-CPMG based experiment, a competition experiment even. And essentially it's ligand observed, so we're just looking at our ligand signals, which also means it's very low on protein consumption. And essentially we're comparing 3 different samples. The first sample, we have our fragment just in assay buffer. We'll get nice sharp lines as we expect from a small molecule in solution. Once we add the protein, if the fragment is binding to the protein, you will see a significant line broadening on the compound signals, especially at higher relaxation times. That's the light green line here. You see that some of the signals are essentially broadened beyond detection. Now for this project in particular, we were focusing or we wanted to target the active site of the protein for which there was a natural ligand available. So in our third sample, what we do essentially is to mix the protein, our fragment of interest, and the natural ligand. And what we see is that if the ligand competes out our fragment, essentially you regain the spectrum of the fragment in solution, indicating that this fragment is competitive to the natural ligand. And the easiest assumption there is there's a good chance that this fragment and the ligand are binding in the same pocket. So we use this experiment to triage our fragment hits and early analogs, pre-select the ones that show this competition effect into our X-ray trials. And once we did that, we experienced essentially much lower attrition rates. That is, the number of compounds we put into X-ray trials started yielding more and more high resolution X-ray structures, which essentially translated to us gaining structural information faster than before. Now, in a very nice sort of positive feedback loop between the 2 techniques, the structure of one of the compounds that we identified after this NMR screen contained the CF3 group. Now, I didn't mention before, but most likely all of you know, this experiment only works for weak compounds. That is, the fragment you are observing in terms of binding and competition has to be in fast exchange, which at some point in the project became an issue because you start getting tighter compounds that are no longer in fast exchange. Now, once we had this structure with the CF3 group of a compound that worked in this assay, we essentially flipped the assay into making a competition-only assay based on 19F. So now we use our fragment essentially as a reporter molecule. We're no longer looking at the new compound, but always looking at our reporter fragment. And we see that in assay buffer, the fragment alone gives us a sharp signal. Now we have a single signal coming from the CF3 group. Once we add the protein, we see that the compound binds by the severe line broadening we observe. This step, by the way, ensures us that the protein is still functional. So it's essentially also a QC step in our assay. We see that the protein is still binding to the fragments in all the rounds we do. And now essentially we test unknown analogs, whether or not they compete out this fragment by regaining the sharp peak in solution. So this enabled us to keep progressing and fast assessing if tighter compounds that were developed and are now in sole exchange are still competitive to our original fragment, and thereby are most likely still binding in the active pocket as we expect. Now, X-ray I mentioned is the first choice to get high resolution structural information in pretty much all drug discovery projects. But sometimes crystal structures, well, we have all the cases where we cannot get crystal structures, but even if we can get crystal structures, sometimes they tell us a story that is not really consistent with all the other data we have backing up the project. More often than not, this is due to crystal packing effects. And that was certainly the case for a project we had with the nucleotide binding domain of HSP70. This was a roughly 40 kilodalton protein that crystallized as a dimer. Here in light and dark gray are two monomers in the asymmetric unit. And we could get a structure with our fragment hit, but the fragment hit was here in purple very nicely binding at the interface between this artificial dimer. We're suspicious about this, of course, and we could change or transfer this project to NMR. And luckily, I already had a great introduction into haddock by Lorna and Stefan. So we did the same. We derived haddock models based on CSPs and NOEs. And this model enabled us to start drawing essentially pharmacophore maps and start assessing which vectors should be explored and let us design the next round of analogs. Having this system enabled for NMR also meant that now we can quickly follow our analogs and compare it to the parent compound in essentially TROSY spectra. And you see, for example, for the peaks I highlighted, that the shifts from the analog are collinear with the shifts with the parent compound, and they are larger in magnitude, giving us an indication that not only the binding site is retained in the analogs and the binding mode is retained in analogs, but also that the affinity is increasing for these analogs, as we also confirmed with other techniques like SPR. So this really enabled us to drive a project forward that was essentially stuck because with the fragment in a clearly artificial pocket, it's impossible to know, or yeah, it's impossible to know how to develop this fragment. Essentially, you just have to try all the different possibilities and see what works. This enables a much more focused path. Just want to briefly mention that this doesn't happen only with fragments. This artificial crystallographic binding pockets. One other example we have very quickly is of a much smaller protein, 20 kilodaltons. In this case, actually with a covalent ligand, where the X-ray structure had clear density for the ligand along a shallow groove on the surface of the protein. But when we looked at this system by NMR, we could see that the major pose consistent by NMR was really tucking in the compound into a much deeper pocket in the protein. And interestingly, actually, there was a second set of NOEs that was also consistent with the pose observed in the X-ray structure, indicating that in solution, even despite the covalent, covalent linkage, this compound exchanged between two conformations. Now, as the last slide, I just want to mention NMR in drug discovery. Well, this is a short, very high level overview of 5 projects we had running in these sort of assay cascades and design make test cycles during the past 2 years. You can see there are a diverse family of targets from inhibitors of epitranscription modifiers to PPI inhibitors to kinase inhibitors. And what I want to impress here is there's no one shoe fits all for this project. You have to design and select which assays and which combination of assays makes sense for each individual target. But for our targets, NMR certainly plays a role in many of the projects. And that can be as an assay to characterize binding of the small molecule to the protein, or to characterize competition, or really as a structural information technique. So to finish, I just want to quickly summarize. I hope I showed you how we use and how NMR can be useful to assess in a very timely fashion the solubility and integrity of compounds. Protein integrity, I didn't really touch upon, but things like the compound precipitating your protein or your protein being unfolded is something that you can quickly check with even with a protein 1D, but also the effect of co-solvents like DMSO. NMR in itself is, of course, a fantastic assay to assess both binding and competition and structural elucidation. So it's really an integral part of drug discovery projects. And lastly, I would like to acknowledge a couple of people, Iso, Johan, and Stefan in particular from the NMR team. Many of the examples I showed come from them. Dennis from our X-ray team also contributed for the project with X-ray that I mentioned briefly. But of course, any of these projects are really a teamwork. So I have to acknowledge essentially the whole of Zobio for contributions that led this project to where they went. And thank you all for your attention. I'm happy to take any questions.
Well, thank you very much. That was great. We've got a few questions. It's always good to hear about the failings of X-ray crystallography, I think. Question from Franz. If you quantify compounds from 1D NMR compared to TSP, do you wait 5 times T1 between scans in order to be quantitative, or do you use a PR reagent to ensure short T1?
Right. So we use it in a more, we usually call it semi-quantitative way. So we don't use PR reagents, and we settled for a couple of seconds recycling delay, which we tested and we found to be a reasonable compromise between experimental time and getting relatively accurate concentration or solubility measures.
Okay, thank you. Vicky Hickman asks, are your scientists trained in multiple techniques, or do you need many experts to work together very closely to move a project between different methods and techniques so you can characterize each target adequately?
Certainly the latter. So most people are experts in a specific area. And whenever we start such a project, we put together a team where we have someone, protein biochemist, to look into protein production and the most efficient ways of doing so. We have SPR specialists, biochemists, structural biologists. So it's definitely a team that comes together with the ideas and the solutions.
Okay, I think we have time for one last one from Christian Griesinger. Maybe you've heard of them. For the structure determination, for the structure determination by NMR, what exactly did you do? I just saw the chemical shifts changing. Also, which affinities did you reach in the competition assays with fluorine NMR? So
for the NMR structure determination, I only showed the CSPs, but for especially the Haddock model, we used a combination of CSPs and intermolecular NOEs. Given the size of the protein we're working with a per deuterated system, so we essentially relied on NH and methyl group NOEs to the compounds. And which affinities did you reach in the competition assays? I'm not sure I fully understand the question. So essentially, we're using the reporter assay, which had a fixed affinity. It was always the same compound, which was in the hundreds of micromolar, if I remember correctly. And then we were using as test compounds to see if they would compete this reporter. They were in the low micromolar range, so single-digit micromolar or so.
Great. Okay. Well, thank you very much indeed. I think we're going to pass over to our